think we are now recording, right? Can you see that, Pete?
We are, yes.
We are. Okay, awesome. Okay, thanks so much again, Pete, for that introduction. Thanks everybody for coming. Yeah, so this is a talk that I developed for as a plenary talk for the Colombian Herpetology Congress this summer. So the first time that I gave it, it was in Spanish. And I also want to thank Dr. Javier Mendez Narvaez, recently of BU and now of the Fundación Calima in Colombia, for inviting me to Cali and specifically for inviting me to give this talk, which is focused on the development of new research methods rather than the development of embryos, which is what I usually talk about, and also on some of the challenges involved in that work. So developing methods has really been a big part of my life and of my career with red-eyed tree frogs, or specifically with their eggs. And it's really been a challenge as well as a pleasure and sometimes a frustration to work with these super cool embryos now for more than 30 years. And they have taught us a lot, opening new pathways for research in biology. And I will share with you part of the scientific process that is behind the discoveries. So sort of the iterative search for ways to ask and answer questions. And this is stuff that is usually kind of hidden in the simplified stories that we tell in our journal articles. Okay, so I study the integrative biology and behavior of tropical frogs, really of their arboreal embryos, but embryos are not a common kind of subject for this type of research. And sometimes established methods for studying behavior don't work. So we know a lot more about embryo development than about behavior. We know more about aquatic frogs than terrestrial frogs. And we know more about temperate species than we know about tropical ones, even though, of course, most of the frogs in the world are tropical. So I will tell you how this new study system generated new questions, how simple observations and experiments led to new discoveries, as well as to other questions that created new challenges in methods development. I'm focused on red-eyed tree frogs, and so that's what I'll be talking about today, but I really think that new study organisms often pose methodological challenges and at the same time can open pathways to new knowledge. Okay, so I began my studies of red-eyed tree frogs at this pond in Costa Rica. And at the time, I knew that terrestrial frog eggs had evolved many times. And this is thought to have been driven by egg predation in the water, but at the time there was really no comparative information on egg predation out of the water. So I wondered, like, what happens to these eggs in the shrubbery? How many survive? What kills them? So I started just doing basic field monitoring to see what happened. And what happened is snake predation. About 10% of the eggs I was monitoring were eaten every night. And I estimated that about 50% were eaten before they were able to hatch. But it seemed that not all of the eggs hatched as soon as they were able to. So I didn't know what happened to the more developed eggs. And at that point, I had no way to distinguish hatchable embryos that had been eaten from those that may have hatched and fallen into the water. And so for context, I did this work on Costa Rica's Osa Peninsula at Sirena Station in Corcovado National Park. And going shopping meant either paying for a charter plane flight to leave the station or walking along the beach for about 8 hours to Karate. There's Karate, where they sold only the most basic essentials like beer and canned sardines. So basically, depending on where you are and what you have access to, something can be easy, it can be difficult, or it can be impossible. I had to wait for the next year to study the survival, not just of eggs, but also of more developed embryos that might have defenses. And this technology, plastic cups and galvanized wires, is very simple. But it really was all that I needed, along with these homemade snake cages. And it was enough to reveal that during the last third of their development, red-eyed tree frog embryos can escape from snakes by hatching. I also did surveys of aquatic predators in ponds. Again, here's a 10, homemade quadrat sampler, tropical hardwood, super heavy. I did simple tadpole predation experiments in plastic bowls in the lab, and I discovered that for tadpoles, being just a few days older and more developed at hatching improved their survival in the water. 2 days makes a difference. Okay, so together, this work demonstrated adaptive plasticity in hatching age, and that embryos can hatch fast enough to escape from snakes. The discovery of predator-induced escape hatching raised new questions, and it created opportunities for new kinds of research. We can use induced hatching both to ask general questions about animal behavior and simultaneously improve our understanding of embryos as organisms that do more than just develop. Okay, so what mechanisms enable this hatching plasticity? How do embryos know when to hatch? What cues do they use? How do they decide? 30 years later, we are still working on these kinds of questions. And I will tell you some parts of this story. But first, let's watch some embryo behavior. Okay, so this snake has already eaten 2 eggs. These 2 embryos have ruptured their membrane, starting the hatching process. And for this one, you will see the whole hatching process in real time from decision through escape. So watch this one. Okay, so here comes the snake. It's jiggling the clutch. It's moving the eggs as it feeds. Okay, this embryo, that did a little wiggle. It's starting its escape hatching process. You'll see a leak where the reflection changes. Now it's made a hole. Fluid is leaking out. It's a small hole. It's going to have to work a bit to squeeze through it. There you go. It escaped. And at the end of my doctorate, I did not have videos this good. This is from new cameras that we got at BU. But I had seen a lot of embryos escape. And it seemed to me that something about the physical disturbance of eggs was the stimulus for this process. And also for my experiments with tadpoles, I had induced hatching manually by just jiggling the eggs and touching them. And so as a postdoc at the Smithsonian Tropical Research Institute, I wanted to test this hypothesis. In Gamboa, in Panama, my mentor, Stan Rand, and really a lot of other people were recording and playing back frog calls, sounds. And it seemed to me, and people seem to think, that working with vibration could be pretty straightforward, pretty much like working with sound. Okay, a little naive at the beginning, that happens. Okay, I started putting egg clutches on various kinds of vibrating lab equipment and other random motors, and no embryos hatched, 0. So then since then, this method, random lab equipment that shake, has worked to induce hatching in a whole bunch of other animals, including insects and vertebrates. So it wasn't a fundamentally bad idea, but it doesn't work in red-eyed tree frogs. I had no evidence that the embryos could respond to vibration per se, but I was not convinced that they couldn't either, right? Maybe it was just that the method didn't work. Maybe not all vibrations induce hatching. And thinking about these gelatinous egg clutches hanging on leaves in the tropical rainforest, it is a very shaky environment. And not all vibrations in egg clutches mean danger. Embryos should discriminate among vibrations, right? So I needed somehow to better understand the natural context of egg vibrations in, different kinds of shaky things, and somehow replicate those to ask a better question. Okay, so first I needed to record vibrations in egg clutches during snake attacks and during rainstorms. How could I do this? So I consulted with engineers, and they recommended 2 possibilities, either an accelerometer or a laser Doppler vibrometer. So laser vibrometer is non-invasive. Its measurements are based on reflected light. But if something breaks the line of sight, it doesn't work. Accelerometers are more invasive. You have to physically couple them to the substrate, and so they could potentially change the vibrations that you want to record. But I thought maybe a really tiny one could work, and also the laser vibrometer cost like more than my annual salary at the time. So I bought the smallest accelerometer available, which is about the size of 2 eggs, and move forward. Okay, so accelerometers are designed to be rigidly attached to solid substrates and to function under low humidity conditions. I had to break basically all the rules of how to use them. But nothing about these clutches is solid. They're basically like a bunch of tiny water balloons stuck together with Jell-O. So I sealed my one accelerometer, in silicone to keep it dry, and I inserted it among the eggs of a clutch, and I gave it to a snake. And I recorded vibrations. Yay. Also learned that snakes eat accelerometers. Some snakes eat accelerometers more than they actually eat eggs. And they make holes in the silicone when they're biting it. And the accelerometer gets wet, it stops working. Fortunately, I was able to strip off the silicone, dry out the accelerometer, reseal it, and it worked again. And me and the snakes and the accelerometer and the eggs repeated this cycle many times. But I recorded vibrations. I recorded vibrations from egg clutches, in snake attacks, and in rainstorms. So then I wanted to know, how do embryos respond to these vibrations? Okay, unlike in bioacoustic research with sound, there were no general established methods for presenting vibrations to animals. A few studies had used playback devices constructed from pieces of equipment designed for other purposes. Others had used manual methods. For instance, Tim Wilcox, super helpful guy, had played waves to water striders using a device that he made from the motor of a chart recorder, these pen things that people used to use back in the day. And he loaned me one. Awesome. But it turns out it was completely too weak to move Aglecnus eggs at all. Okay, so I talked to engineers again, and they recommended this mini shaker. It has a much stronger motor that moves this platform up and down. So I needed a way to attach it somehow to the eggs. I made this interface out of a bent piece of wire. I had a cheap off-the-shelf amplifier from Radio Shack, just one laptop computer, no way to both play vibrations and record them at the same time to verify what I was actually playing. I picked a playback amplitude by eye just by looking at it. Seems reasonable, right? And these definitely were not high fidelity playbacks. They were very low fidelity playbacks. Nonetheless, they were good enough to ask the embryos a question like, is this scary? Or is this more scary than that? I played vibrations recorded from snake attacks and from rain to embryos, and they distinguished between them. Wow. Okay, this was super exciting. And it opened new possibilities to ask questions about how embryos use vibrations to assess risk. For instance, do they use temporal pattern information? We could do that. I could do that. I edited my recordings manually, moving pieces of silence around to either group or divide vibrations. Once again, the embryos distinguished between them. They hatched more in response to patterns that were more snake-like and less in response to patterns that were more rain-likes. And again, these playbacks were not high fidelity, but they revealed completely new things about the behavior and abilities of these embryonic frogs. So some biologists had thought that these embryos evolved specifically to hatch in response to rain, and that their response to snakes was just incidental, really that any movement at all would stimulate hatching. That was wrong. The embryo behavior was more complex and it was more interesting. Okay, so with this work, I got a job at Boston University. I got a vibrations engineer, Greg McDaniel, as a collaborator. And then together we got an NSF grant and we recruited a co-advised PhD student, Michael Caldwell, who was interested in vibrations. And we all, three of us, went back to Panama to the Smithsonian. And we bought more accelerometers. We made more vibration recordings from egg clutches in the field and also in the lab. And we learned that not all accelerometers perform equally well, even the same model from the same company, and also that our testing conditions revealed production faults standard engineering tests do not reveal. And understanding and solving this problem and convincing the company that made the accelerometers that this was real cost us 5 months. 5 months of my first NSF grant, paying like 3 people to be in the field. Okay, but we moved forward with our studies of temporal patterns, using synthetic stimuli to map embryo responses to vibration durations and silent intervals. And again, the embryos showed strong discrimination among patterns. So a limitation of this initial interface that I had made from bent wire was that some eggs were close to the tines and some eggs were further away. And in large clutches, this was definitely worse. So my collaborators, Michael and Greg, made a better interface, gluing tines from a poodle comb into an acrylic plate. And this was awesome. This really improved our control of egg motion a lot, and it let us work with a larger range of clutch sizes. And this let us use temporal pattern manipulations to ask more questions. For instance, we learned that embryos use a flexible information sampling strategy, and they base their decisions on less information when collecting that information is more costly, takes more time. We also wanted to know if and how embryos use frequency cues in vibrations, right? Our recordings from snake and wasp attacks were dominated by low frequencies, and rain vibrations include a much broader range of frequencies, higher frequencies. And this suggests that embryos should hatch more in response to low frequencies and less in response to higher frequencies. To actually test this, we would need much better control of frequency in our playbacks. Okay, so let me explain. Here's a set of equipment for playing vibrations. We send a signal from a computer through a set of gear, series of devices to vibrate a substrate, okay? And then we can record the vibrations, passing them back through another set of devices to another computer, and there's our signal again. Great, except it isn't. It is not the same signal when you get it back, because every piece of this equipment filters vibration frequencies. The solution to this is to compare the returning recorded signal with the original signal or with the signal that you desire to present, and to compensate digitally to generate the desired vibration in the substrate. Okay, so this is what we needed to do. Thinking about the substrate, vibration transfer depends on the physical interface between the shaker and the substrate, and also on how the sensor is connected. And for consistency, the recommended engineering methods are either screws or glue, but both of these depend on having a solid substrate, right? For instance, other scientists, biologists who work with communication signals that are transmitted as vibrations in plants often connect their devices to a plant, and they perform a series of digital recordings and adjustments to optimize playbacks for a very specific location on the plant. And then, without moving or disconnecting anything, they can place a series of different test animals in that, on that leaf or whatever, that particular part of the plant, and they can ask them questions. But with a gelatinous structure, these connections are inevitably going to be less consistent. And for every single playback, we needed to connect a new egg clutch. Also, our animals are always already inside the structure that we're going to vibrate. So we cannot optimize playbacks for individual clutches, right? If embryos hatch, the physical structure changes, which changes the vibrations. If they don't hatch, we have changed their experience, right? It's not the same to present a stimulus a second time to the same animal. So because of these things, perfect playback fidelity is impossible. Realistically, our goal is just adequate playback fidelity. And the requirements for adequate playback fidelity depend on the study system, and they depend on the question that you're asking. Okay, so let's think about vibration properties that we can measure and control. So these are frequency spectra for 2 raindrops are slightly shifted vertically, and looking at their peak amplitudes, there's a difference in frequency, right? Higher, lower. Looking at the acceleration over time and the waveforms of these 2 drops, they have different fine scale temporal patterns, and they also have different acceleration amplitudes. And we can assess temporal patterns at different scales. This shows a longer period of vibrations from a parrot snake attack. Okay, so for frequency measurements, at the low frequencies that matter most for embryo behavior, our accelerometers perform quite consistently across different attachment methods. Although at higher frequencies, attachment method really changes their response. For amplitude measurements, these little accelerometers are designed to record one axis of movement and have very low sensitivity in other directions, which means that axis alignment changes the measured amplitude. So these 2 recordings are from the same stimulus, played at the same amplitude to the same egg clutch. I just adjusted the accelerometer alignment slightly between them. So what does this mean? Our recordings give us excellent measures of the temporal pattern of vibrations, but they inconsistently underestimate vibration amplitudes. Even like within the same recording. We cannot control the directionality of snake movements in attack. We cannot control the directionality of raindrops in storms. So their measured amplitudes are almost always going to be less than their real amplitudes, right? And how much less varies even within a single attack or a single storm. Okay, so before you get too worried, at this point, I want to remind you or tell you about the difference between stereotyped communication signals and incidental cues. So communication signals like this are pretty consistent, and receivers are very sensitive to variation in the details. It matters to them, and so we need to control them in our playbacks. But incidental cues like this snake attack vibration, they're super variable, which means that prey that have to distinguish these from background environmental noise need a response that's going to be robust to a lot of variation. So here are 2 attacks by different species of snakes, the parrot snake, and this is going to be a cat-eyed snake when it comes into the frame. Each species of snake has its own feeding habits. It has its own movement patterns, but also even from one attack to another by the same individual snake, there's a lot of variation in the egg clutch vibrations. And then wasps eat the eggs, katydids eat the eggs. All of these different predators make different vibrations. And in spite of this, all this vibration, the embryos can distinguish between benign and dangerous.
Where?
And they show actually very similar escape rates in attacks by 4 different species of snakes.
It's a
that have
cooler?
vibration patterns.
Oh, I'm watching a talk and I have office hours. Maybe I can go.
People online maybe mute. Okay. All right. So to test embryo responses to frequency, we first measured the transfer function of all of our gear and the frequency compensation that we needed. And we did this in Boston without eggs, and it worked great. Then I went to Panama, and when we connected the gear to the eggs, everything changed. It's me and Michael in Panama. Also, every clutch is different. Different sizes, different shapes, different vibrations. It took a while to get to this, but our solution was basically just to measure the transfer functions for a bunch of different clutches, different sizes and shapes, and take an average. We decided that this hopefully would be good enough to start testing how embryos respond to frequency. It is definitely not perfect for any individual clutch, but with a structure that varies among exemplars and changes during the course of experiments, the frequency fidelity of playbacks is always going to be limited. And in fact, it was good enough. We learned that embryos do use frequency information. We had to buy a bigger shaker for adequate playbacks at the lowest frequencies, and it turned out that these low frequencies induce the most hatching. And this is consistent with their dominance in recordings of attack. But okay, what about these high frequencies? By playing rain recordings and the same recordings filtered to remove higher frequency energy, we learned that removing high frequency energy actually increases the hatching response. And this is work from Michael Caldwell's PhD. Okay, so we learned a lot from these tine playbacks to egg clutches, but we did them in a narrow developmental period when embryos are highly responsive to mechanosensory cues and also highly discriminating. So we could set up experiments, we could answer questions using a reasonable number of egg clutches. But younger embryos are less responsive. Older embryos eventually become impossible to set up for experiments without inducing hatching. So we knew that development changes behavior. We just couldn't actually use this system to ask questions about that. And embryos change a lot during the period when they're able to either hatch or remain in the egg. And this matters. We know that from predation experiments where the escape success increases dramatically from 4 to 5 days. But how does development change embryo behavior? What can they do differently at different developmental stages? And what are the mechanisms underlying those changes? And also, why does development change their behavior? Is it just that they're developing new abilities? Or are there adaptive changes in how they respond to the exact same information due to changing costs and benefits of life inside and outside the egg? So tine playbacks were absolutely inadequate to answer these questions. So Greg and I decided to develop a new system for presenting controlled motion signals to groups of individual eggs. We wanted to put them in some kind of standardized structure, let them develop, and then connect them to our shakers without touching them. And we wanted to present tactile cues independently, either alone or in combination with motion cues. We had some requirements for this new system we were going to develop. In terms of the biological requirements, we needed that embryos would develop and hatch normally from the system, and also that they would tolerate connection to the shaker without hatching. From the engineering or mechanical side, we needed the system to actually work under tropical rainforest conditions. We needed it to transmit vibrations well, robustly, and to enable easy, reliable setup for experiments. I wanted undergrads to be able to do this. Okay, meeting this combination of biological and mechanical requirements took many iterations of design and fabrication in Boston, and then testing and modification in Panama, learning from every failure. Our motto for 3.5 years was solve a problem, reveal another problem. Okay, before I tell you more about this process, I want to say for investing in methods development, context matters. At this point, I had worked with agolignous embryos already for 21 years, and I had done many playback experiments. So I knew they were a great study organism, and I really thought that this investment would be worthwhile. I also knew the embryos well enough to believe that I could figure out an effective rearing system for isolated individuals. And also, Greg and I had an established and productive collaboration. So we were able to enter into this process with really no idea how long it was going to take. Or possibly we were just like optimistic and stubborn. These are not mutually exclusive. Also, my lab had other projects underway at the time, including work on hatching mechanisms and studies of parent embryo interactions in glass frogs. And finally, I want to say that even with a more controlled playback systems, simple methods like manual egg jiggling are still useful. My student, Majo Salazar-Nichols, is actually right now in Panama using egg jiggling in combination with drug treatments, manipulating neurotransmitters and their receptors to study the neural mechanisms underlying hatching enzyme release. Okay, so in 2012, we started working on standardized egg holding structures, drilling cylindrical holes into pieces of ABS plastic. And we tried different sizes and depths of holes. And it was actually surprisingly difficult to optimize for both healthy development and hatching success. But okay, fast forward a little, we did manage this by carefully controlling egg hydration. Then we worked on the shaker interface. First, we tested metal to magnet couplings. Stronger connections caused hatching in setup, and weaker ones rattled during playback. And we tried many variations of this, looking for the sweet spot that would work, but it didn't exist. All right, the next year, we tried a screw-based coupling, and the vibration transfer was great, but mounting the egg holders took far too long, and the embryos hatched way too much. In fact, it was so bad, I don't even have a photo of this system. Okay, so in Panama, I modified this interface to use spring clamps. The connection was robust, great vibration transfer, and it worked really well with 5-day-old eggs. But at 6 days, they still hatched too much in setup. Then I used heat molded plastic to make the first asymmetrical interface. So the egg holders had these flanges that fit into a slot on one side of the interface, and I used a spring clamp on the other. So I thought this just might work. And again, it was a robust connection, with good vibration transfer. Why is that not playing? And finally, I could set up older eggs for playback. And we started testing this with a wider range of playbacks. And then we learned that at some important frequencies, at reasonable amplitudes, come on. The egg to holder coupling failed. Solve a problem, reveal another problem. And back to Boston for redesign. Okay, so my engineering collaborator, Greg, suggested angled cones so that gravity would help to hold the eggs in place and the tadpoles could slide out the back. Also, any egg size would fit. So we made a set of prototype variations from 3D printed plastic. And back in Panama, I very quickly learned that 3D printed plastic wicks water out of eggs. We tried spraying them more, but the cones trapped water and embryos died of dehydration while others drowned in the exact same structures. It was basically impossible to keep them healthy in this thing. But some elements of this system did work well. The connection system was our first cone and clamp coupling. It was very easy to set up and it was robust in playback. And it was super convenient that all egg sizes fit. So I hand drilled and carved some funnel-shaped spaces into ABS plastic blanks that were cut to match this interface. And the embryos developed well and were able to hatch out of them. But in playback, eggs fell out. This was like way worse even than with the cylindrical spaces. Okay, then Greg suggested we rotate the whole system 90 degrees so gravity would help us again. And after fixing the interface with more thermomorph, I tried it. And I want to say that this is actually kind of a scary thing to try because we did not want the torque from this cantilevered mass to mess up our shakers. And so we cautiously tried different kinds of foam supports and I tested shaker performance and we found a configuration where we thought that the shaker worked well. Now, more recently, with another supposedly very similar shaker, and an engineer from that company was like super, super concerned about this setup. So just like not all accelerometers are equal, not all shakers are equal either, despite their engineering specs. Okay, so finally, after 3.5 years of work, we thought that we had a set of methods that were good enough now to ask our new behavioral questions across development. We call these egg holders egg trays, and the playbacks are tray playbacks. All right, and with this new system, I started to test the adaptive ontogeny hypothesis. I wanted to know if developing embryos change the way that they use cues as the costs of decision errors change. For embryos sensing vibrations that might be from a snake, development doesn't change the risk of staying in the egg, right? It's never good to be eaten by a snake. But for younger embryos, they face a high risk of predation in the water, so they have to balance these risks. Older embryos, close to spontaneous hatching, induced hatching really has little impact on their survival. So if vibrations are costly or time-consuming to assess, they should just hatch rather than just wait around and see if it's a snake. Okay, so we designed stimuli based on our prior results with younger embryos, and we played them at 2 ages. We included a positive control that elicits high hatching, a benign stimulus that is fast and cheap to assess, to test whether older embryos just fail to discriminate or hatch with any vibration. And then our third stimulus elicited a little hatching of younger embryos, but the pattern delivers information slowly. So it takes more time and more risk to assess. We predicted and tested that over development, the response to the slow pattern would increase more compared to the 2 controls. Okay, so this video shows older embryos in a slow pattern playback. The stimulus causes very little hatching of younger embryos, but you'll see in a minute that with these older ones, a bunch hatched. And we tried that twice with different discrimination controls, and the results were very clear. Older embryos do assess risk, and they do discriminate between signals, between stimuli, but the way that they do it is different. It changes with development as we predicted. So trade playbacks had taught us something new. right? They taught us something that we couldn't learn with timed playbacks. I was very excited about this. Our reviewers were not. This paper was rejected 4 times. The reviewers did not like the playback system. They recommended a bunch of things that we had already tried, which did not work. They wanted things that seemed unnecessary or impossible, and they really could not see the value of this work. The first reviews were helpful, the later ones, not so much. Okay, then, same paper, another group of reviewers loved it. They saw it completely differently. Maybe they worked on different systems, maybe they thought more broadly, I really don't know. But I was so happy to finally publish this because by then, my student Julie had data for like several more papers with the same playback system. And I was like, we cannot have her papers rejected 4 times. Okay, we wrote a detailed methods chapter on approaches to studying mechanosensory cute hatching. And so far, Julie has published 3 more papers using tray playbacks without this kind of difficulty in review. Okay, so we know that animals use incidental cues as much or more than communication signals, but we very rarely know how. But for vibrational cues, we can learn. In some ways, vibration is harder to study than sound, although I think it's way easier now than it was 20 years ago. But the variation in incidental cues means that even, well, if the information is important for the animals, receivers are going to need robust decision mechanisms. And that means that our methods for studying them don't have to be perfect, right? They just need to be good enough. And that makes it easier to work on new systems. So I want to emphasize that playback fidelity needed for an experiment really depends on the question being asked. And others have said this before for other kinds of behavioral experiments. The key is to have appropriate controls and careful interpretation. Every improvement in methods in an experimental system, it takes time, it has costs, it makes sense to do the easier things, which might be less precise or less controlled first, to see if more difficult things are actually worthwhile. With vibrations, we can actually learn a lot from simple manual disturbance, starting with cue modality. A little refinement allows for basic tests of discrimination, and then with better playbacks, we can ask more kinds of questions. A lot of embryos, not just frogs, use vibrational information, and I am hoping that others will build on our work for studies of embryo behavior in other species, and also use embryos to learn more broadly how animals use incidental cues. Okay, I want to tell you one more story, a short one, which also started with incidental, simple observations. Okay, so these embryos have beautiful external gills, and I wanted to photograph them outside the egg. But every time I prepared an animal for photos, they look different. This is me as a PhD student, right? Now, they look very different. What was going on? I made a time series of observations starting at hatching, and I saw that external gills regress very rapidly. And then I manipulated oxygen availability, restricting it in the water and increasing it in the egg. And these manipulations changed gill size and perfusion. And the contextuality of gill regression suggested to me that these things have a physiological function. I thought that these elaborate external gills and the plasticity in their regression might facilitate prolonged development in the egg and plastic hatching timing. Okay, a manuscript reviewer wrote, "It is not possible to see blood circulation in the gills, the external gills of anurans. We already know that these are vestigial structures with no physiological function." And I suppose the reviewer worked with a species like some toads, for which these things are true. But frogs are not all the same. In agalicnus, it's easy to see blood circulation in the gills before there's even much hemoglobin making it red. And at the time, I did not know that I could challenge this rejection and explain to the editor that the reviewer was wrong. So I sent it to another journal, which published it. And I thought that external gills and access to oxygen could be important for development and could be important for hatching timing. So I continued to study this, and I published a series of empirical papers. But I felt like an imposter doing this work. I had no training in respiratory physiology. Nobody in my lab studied it, but the embryos motivated me to continue. And I continued to learn. I continued to talk with colleagues who knew more about respiratory physiology. And then I was invited to present at this symposium and to write a review article. And the journal that had rejected my first paper on the external gills of agalicnus published my 6th, which was an undergraduate thesis in my lab at BU. And so this continues to be a productive area of research for my lab. We just published a paper using hatching in response to hypoxia, as well as egg jiggling, to study the ontogeny of risk assessment and hatching performance. These embryos are different ages, younger, older, and you will see that it is harder and it takes longer for the younger one to hatch. And also, Majo Salazar-Nichols is in Panama right now studying the mechanisms enabling embryos to sense oxygen level, which is another function of these beautiful gills. It's going to get out, don't worry. You know, and the difference in this is also telling us something about hatching mechanisms underlying how they get out and how it changes developmentally. Also something Majo is working on. Okay, so most of our established biological knowledge is based on a small subset of the diversity of life. It's an important foundation, right? We know a lot, but our knowledge base also includes biases, and it includes overgeneralizations that can block scientific progress, that can limit our understanding. But, you know, frogs or, you know, whatever animals or bacteria or plants or whatever you work on, they just do what they do. They do not care what we already know. And what they do varies. And I think that studies of a greater diversity of species can help us overcome some of these historical biases to build a broader and better understanding of life. Our job, sometimes our challenge, is to not limit ourselves in advance to seeing what we already think is possible, but to just clearly see all of the diversity that exists in nature and try to understand it. I want to thank the agencies and institutions that have funded and supported our work, all of the frogs that we have studied in their different life stages, and all of the people, in particular, my students and collaborators who have contributed so much to improving our knowledge. And I've also shared my joys and my frustrations over the years. It has been such a pleasure to learn new things together with all of these people. Thank you all for listening in person and online, and I will be happy to answer questions in English or in español. Okay, and I am gonna just, let's see, stop the sharing and so that I can see online people as well. Yes.
So I've worked with the spatial toads in the past, and whenever they change their development to escape these ephemeral ponds, there's these long-term consequences of lower biomass and like slower development into adulthood in general. Do you see the downstream consequences of like high escape early versus high escape later into adulthood, and does that carry on?
Yeah, so the question comes from someone who has worked on spadefoot toads and understands something about the longer term consequences of accelerated development to escape a deteriorating environment. Do we see that in agalicnus? So absolutely, there are trade-offs around hatching timing. And so we have measured short-term costs in terms of higher predation and sometimes just lower viability in the water over the first few days. It seems to be something concentrated in the first few days after hatching. Then there's also compensatory growth. So the preemie ones catch up, and then after a few days, like you can't tell them apart, and we lose that differential mortality. In some experiments, but not all, we see a secondary period of differential die-off shortly before metamorphosis, which is a high stress period. This is maybe kind of related to we see in mammals, and it's a public health issue in humans, that preemie babies have catch-up growth, and then later at puberty, they can, some further problems can emerge associated with that. We have not followed them like into adulthood or longer. They take a long time to grow up, and usually we are not in Panama in the dry season. But I will say that I have a new collaborator, Sally Bernardina Serafin at Trinity University in Hartford, who is a neuroscientist and is really interested in effects of early life stress and trauma on brain development and on later behavior. And there's a lot of importance of this for humans, and has just started breeding red-eyed tree frogs in the lab, and is interested in potentially using, embryo stresses and induced hatching to look for a sort of longer term neurobiological differences in these animals. So we don't know about those kinds of costs or downstream consequences, but there might be, and we will, she will be finding out about that. Yeah, good question. Thank you. Any other questions? Pete.
Other people in other criminal businesses starting to adopt these migration?
Well, certainly there's like a lot of vibration playbacks that have been done to lots of animals. I would say there's like vibration playbacks to study communication. There's like far more of them. And incidental cues, not so much. And so I don't know of anyone who's using, like, say, our the egg tray method stuff is pretty new. The biotromology book, our methods chapter, just came out in 2022. So I think that like that, it was delayed by the pandemic, that book publication. And so I don't know yet, but I will say that when I was in, so as I say, I gave this an español in Colombia in August, and there were a bunch of people at the Colombian Herp meetings who of course have access to all kinds of cool frogs, right? Who were very interested in the vibration stuff. I talked to several students who are like interested in doing vibrations PhDs. And so some of them maybe would work on eggs. This like is very obvious and easy transfer to like other species of terrestrial frog eggs. I don't think it would take much adjustment of the system for that, but then I mean, could be modified in different ways for really any kind of egg. And there's lots of eggs that have vibration cued hatching. So I would love to see this expanded for learning about how animals use incidental cues. Yeah. So I hope so. Any other questions? Anybody online have questions? Okay, well, thanks everybody for coming.